Yeah. Being able to be your true self is one of the strongest components of good mental health, said Dr. Lauren Fogel Mercy. Good afternoon, everybody. Indian Women Scientists Association was established on July 19th, 1973. Now there are 11 branches across country. In order to take science to society using science and technology for the upliftment of the not so privileged class of society and for women, IWSA has been conducting number of science programs. To name few science programs: Science Nurture, Lecture Workshop, Science Refresher Courses, Research Projects, Popular Science Lecture Series, etc. Under Popular Science Lecture Series, IWSA Bangalore branch, in association with Department of Genetics Indian Academy Degree College Autonomous, organizes webinar on models and genomic measures of psychiatric disorders supported by BRNS DAE. I cordially welcome all the participants to the Popular Science Lecture. Today's resource person of the webinar is Dr. Ritika Sir. Dr. Ritika's talk will cover the models for studying psychiatric disorders with a special emphasis on stem cell models. The talk will also discuss what they have learned on the genetic and genomic architecture of these disorders, what these techniques have and have not revealed about the enigma of mental illness. All that being said, now I take this opportunity to welcome our Honorable Chairman, Dr. T. Somshekhar, Indian Academy Education Trust and neurobiologist as well. I also extend my welcome to an esteemed secretary, Mrs. Bharati, Indian Academy Education Trust, respected COO, Dr. Srinidhi K. Parthasarathi, Indian Academy Group of Institutions, Reverend Principal, Dr. E. Jerome Javier, Indian Academy Degree College Autonomous, and Dr. Savitri Bhatt, convener of Popular Science Lecture, Bangalore branch, and also welcome the members of IWSA, participants from various universities and research institutions, colleagues of IADCA, and dear students. On behalf of IWSA and Department of Genetics, I am glad to welcome Dr. Ritika. Dr. Ritika, educated from University at Buffalo, the State University of New York, US. She is working at National Institute of Mental Health, popularly known as NIMHANS, as a research coordinator of the ADBS lab. Dr. Ritika's lab studies the genetic and cellular basis of psychiatric disorders as OCD, schizophrenia, bipolar disorder, addiction, and dementia. Dr. Ritika carried out a number of research projects. Currently, she has been working on the project entitled Dissecting the Biology of Lithium Response in Human-Induced Pluripotent Stem Cell-Derived Neurons from Patients with Bipolar Affective Disorder, which can have significant positive societal impact. She has a number of research publications in the peer-reviewed journals to her credit. To mention a few: high incidence of leukemia in large animals after stem cell gene therapy with a HOXB4 expressing retrovirus vector. An audit and insight from clinical exome sequencing in psychiatric genotype-phenotype correlation. Migration, a tool for analyzing cellular phenotype for neural stem cells derived from bipolar disorder patients, etc. Dr. Ritika also volunteered with India March for Science, Bangalore, Indian Scientist Response to COVID-19, the Bangalore chapter of 500 Women Scientists, and inclusive. Dr. Ritika is very passionate about science communication, on which she has been conducting several workshops. She has also contributed articles to several news outlets like Deccan Herald, The Wire, Times of India. We are very happy to have such an eminent personality amidst us to instill young minds through giving insights on models and genomic measures of psychiatric disorder. Welcome, Dr. Ritika. Participants are eagerly waiting to listen to you. Over to you.
Thanks very much. Thank you for inviting me, and thank you for the generous introduction. I wouldn't call myself eminent personality, but thank you for the honor.
Thank you.
Okay, so what I want to touch upon today, since most of our audience, I'm told, are students of genetics, to talk to you more about what we know, what genetics or studies of genetics and genomics, which is one word for your entire DNA content, what that has told us about psychiatric disorders, and the limitations of it, what are still the open questions of it, and how do we go, more importantly, from what we know from genetics to to a disease. So essentially that genotype-phenotype connection. So what is genome exactly? Let's start there. Genome is one word for all of your DNA content in a cell, all of it. But the big question in genetics always has been, how do we go from product of our genes? But then there is a lot of open questions there, and a lot of misunderstanding about how much do genes contribute to what we do see in terms of phenotype. I mean, it's a well understood fact, at least in theory, that genes interact with the environment, which is your lifestyle. Are you someone who's stressed or not? What kind of diet you have? What other lifestyle factors? Are you active or not? For example, what chemicals you may be exposed to, so on and so forth. All of that goes into then finally producing a phenotype. A phenotype can be a visible trait. A phenotype could be a behavior. All these are pieces that form that puzzle, but then the connection of how each piece connects to the whole puzzle or to other pieces of the puzzle isn't always clear. One thing we do know is that, so in our genetics classes, we hear about, or we learn about mutations and so forth. But one thing to know is that it's not always because if a mutation is linked to a disease, it's not always a straight connection. If you have this mutation, you'll have this disease. Not necessarily. So DNA is not destiny. It's a blueprint, it's a map, it's instructions. But then a lot of what happens beyond those instructions is how those instructions are read, how they're followed, how they're implemented. And all of that goes into finally producing what is visible, which may be a disease, which may be a behavior, the entire spectrum. So when it comes to human traits, phenotype, when we're talking about humans, so when we start learning genetics, we learn Mendel, first of all, pea plants and the traits of the pea plants. That's more like the ABC of genetics. Yeah, that's just the building blocks. That's how he made sense of it. But human phenotype works a whole lot differently. We can have similarities to what Mendel discovered, where you have alleles, and then which alleles you have is a direct reflection of what phenotype would be evident. So an example of that is hair color. So this is one gene, and depending on which alleles, which version of that gene somebody has, if you have both copies that are unaltered, you will have black or brown hair. But if you have both the copies are altered, you'll have red hair. So that's one end of the spectrum. One gene, one phenotype. You have the other end of the spectrum, height. So there you have over 700 genes. And that's just genes. We're not even talking about the effect of environment, what kind of diet somebody gets, what kind of environment they're exposed to. Just the sheer number of genes that are involved in influencing that phenotype is over 700. All of those will have their interacting partners, and then combined activity of all of those will somehow translate into this phenotype. So again, not a direct, this gene or this version of this gene, therefore this phenotype. I will just give a quick overview of what the different variations are. Some of you may have already learned about these in your class. So for example, we have this original sequence. This one example, the sky is blue. And then what are the different ways that this sequence may be modified? So you may have one character switched out with another. This is called single nucleotide polymorphism, or SNPs. A lot of our talk today will talk about SNPs. So, which is what this is, just a single base, a single nucleotide is changed to something else. You can have changes that extend beyond one nucleotide. So that's what are called indels, which is insertion and deletion. So you can have two or more nucleotides added or deleted. So in which case it's either an insertion or a deletion. There are also variable number of tandem repeats, which is the same entity here repeated a bunch of times, and copy number variants. These are structural changes in the, so whereas here, these are sequence changes, CNV are structural changes. So you have different pieces of DNA either there in multiple copies. And all of these are comparison to, here it is called original sequence, but when we're doing sequence analysis, this would be called a reference sequence. These days, it's computerized databases that can be accessed from anywhere in the world, and where you would have the reference sequence, and then once you have a sequence of your choice, you can match that to the reference sequence and see how well it matches or where any of these changes are, and also what kind of changes. So this is overall what we'll talk about today. As I said, what does genetics tell us about a disease, and how do we use that, the information from genetics, to then understand a disease or treat that disease. So the interesting thing about psychiatric disorders, which also makes it challenging to study, to understand, is that they impact higher cortical functions. So cortex, part of your brain that regulates cognition, it processes information to make sense of the world. The parts of the brain that regulate mood are different. The cortex regulates those. So your mood, your behavior, all of those are functions. One or more of these functions are impacted in psychiatric disorders. And a lot of these are unique to human brain. So studying them makes it all the more difficult. What you have in a disease is a visible change in a person with the disease. How that person relates to what's going on in their brain, whole different ballgame, and extremely challenging then to understand. This in contrast to a structural problem in any other organ, where you can see, well, this injury and therefore this change. You're not able to, you have this injury, so you're not able to move a limb which is injured, very straightforward. And because brain is not accessible, that makes it all the more difficult to understand how changes in brain are leading to changes in behavior and even disease. So what do we know? We know, I hope this is visible. Let me enlarge that a little bit more. Okay, so at one end of it, you have genetic architecture, which is what is the DNA sequence. So in the more recent, which is last 2 decades, 2003 was when the human genome was sequenced. So from that time, we're talking sequencing at genome level, at large scale, global sequence of the entire DNA of a cell. From that, so from learning about what a sequence is, a lot of times we can, from sequence, we can relate to, for example, if you're doing a case control study, you have a group of individuals, some of them have that disease and some don't. And you find, so you do DNA sequencing, genome sequencing to find out what are the differences in their DNA that are responsible for this phenotype, phenotype difference, which is presence or absence of disease. So you sequence the DNA, and at that point, the goal is to find out, can we narrow it to this one or more of what are called causal variants, a variant that you can link to. If you have this variant, you're more than likely to have the disease in question. So that's your genetic architecture. The sequence may be different, which may be contributing to the difference in phenotype, presence or absence of disease. So you go from a DNA site that is different to identifying the gene that is the source of that difference. but no gene or no protein that the gene codes for acts alone. So then how do we go from that gene to then the disease? We try to identify the pathways. What are the other genes that this gene talks to, either functionally or physically? So that tells us about the pathways that may be affected. What are the cells that this gene and this pathway are active in? That will make a difference. And at what point in then development does this come into play? From that, you form circuits. So pathways are inside different proteins talking to inside cells or maybe between cells. Circuits are you're going across brain regions. So essentially, your goal is to be able to complete this whole path, which isn't always possible, especially when we're finding out new variants. It can take a while to go from here to here. And from all the sequencing that has been done so far, we don't even know this whole thing for every variant that has come up from DNA sequencing. So once we find out what a variant is or where it is there, where it is present, it can be either in a coding part of the genome or in a regulatory part of the genome. So regulatory parts are, most of the genome is regulatory. Only 2%, as most of you may know, codes for proteins, which is to say that 98% of it does not. So if a variant that is known falls under that, then this takes a whole different meaning. Because going from because unlike the coding part of the genome, there is a lot that the regulatory region that we don't know about. The genes, for example, follow a set features. So there is a start site and an end site, and exons have what you can call landmarks: the start of an exon, end of an exon. So those are molecular landmarks, you can say, for beginning and end of gene and beginning and end of exons within those genes. So if a variant falls in that part, it can, relatively speaking, it may be straightforward to go from genes to pathways and then forward from there. But regulatory regions don't always follow that same logic. There isn't a start and end that's as clear. And they may be present anywhere in the genome. So we know where a gene is present, gene X on chromosome Y, but regulatory elements that control that gene may be present anywhere, may not even be on that same chromosome. So that makes it sort of challenging to figure out. And I'll talk some more about the techniques that are used to identify regulatory regions and genes. So the point here is simply to say that when there is a difference in DNA sequence that's identified, that in a gene or in a, so those are different ways that sequence that we go forward from sequencing, depending on where that change in sequence is. So once we find out, okay, we have a SNP, a change in DNA sequence, the next thing is then to see, well, how much of a difference does that make? I mean, all of us have the same genes, but then barring identical, most of them are of cases and controls, it can mean that it's linked to the disease. So that we have on the y-axis here, which is the effect size, effect of that variation on the outcome, in which here it is the presence or absence of disease. So when there is a variant, it can have a very low impact on the development of disease, or a very high one. That variant could be very rare in a population or very common. So outcome of a variation on a phenotype, in this case a disease, is a function of both of those things. How common is that variant in a population? And then how much of an impact does, when this variation is there, how much of an impact does that make on a disease. So on one end, you will have rare variants. So variants that are very rare, and also when they're there, they make a very small impact. So when that is the case, it makes it rather difficult to prove a correlation between, because you have this variant, it has something to do with the disease. You have the other part of it is that the variants are rare, but when they are there, they make a big difference. So that's your Mendelian diseases. When a variant or a mutation is there, it increases the chances greatly of somebody developing the said disease. And then you have an entire spectrum of variants classified by their frequency and how much of an impact they make. This other end is the common variant. So these are common is there that said variant in sequence is present in 5% or more of population. So that's said to be common. And rares are less than 1%. So you have variants that are common, but they have low impact. So there again, that's a different kind of a challenge. How do you, it's a common enough variant, which means a lot of people who have the variant do not have the disease. So how do you go from demonstrating that this variant has anything to do with that disease? And then you have variants, this are very few in number that we've learned so far, is that variants that are common enough, but then have a large enough impact on disease when present. So I'm going to use Alzheimer's disease as an example. So Alzheimer's disease, like a lot of brain diseases, has genetic basis. So on one end, you have these variations in these three genes, APP for amyloid processing protein, precursor protein, sorry, and presenilin, PSEN1 and 2. When anybody has, there are certain mutations in these genes, when those mutations are present, which are, they're rare, so it does, if you look at patients across the board, this may be in less than 5% of cases on average, who will have these mutations. But when they do, they greatly increases the chance of somebody developing Alzheimer's disease. So that's these three genes right here, which is this end here. So rare alleles, but when they're present, they greatly increase the chance. Other than these, so that these, like I said, is less than 5%, which means most of the people who have Alzheimer's disease do not have these. So then what is genetics involved there at all? So for that, when analysis was done, we found a whole range of variants. So you have variants linked to all these genes that vary a lot in their frequency. These are extremely rare and also have a smaller impact, to these ones that are fairly common, but then their impact is still low. So then how does this translate all of these genes into Alzheimer's disease? That is still being worked out. So what we do have is genes related, a list of genes that may ultimately be working together in pairs and combinations. That is still being studied. So, but this is one end. Like we have a sort of limited list of genes that are linked to a brain disorder. On the other end is schizophrenia, which, as you will see from here, this is what's called a Manhattan plot. This is every, when you look at variants across the genome. So on your x-axis are chromosomes, 1, 2, 3, so on till chromosome X. And every dot here is one variant, okay? And so this is, the y-axis is the probability, so p-value, which is to say that what are the chances that a variant is actually linked to the disease. So when you plot all the variants, all the so you've sequenced the entire genome, you found out all the variants, you put them all in like a scatter plot, all of them here. Now, the ones in green are the ones that are significant. So the red line that you see here, this is the significance threshold. Any signal, any variant that is higher than this, there is a statistical chance that that variant may be related to schizophrenia. So as you see from this figure, that the genetic links to schizophrenia are spread all across the genome. Last count was over 100. So that's a lot of variants to then link to, well, how do we go from here to the phenotype, disease phenotype? Ways that can work. So you were here at this end. You have sequence variations, differences in DNA sequence in individual genes. So those are your SNPs. They can also be structural variants. So there is one in particular on chromosome 22. When that happens, people develop schizophrenia, but like was the case with Alzheimer's disease, when you have people who have this deletion are very likely to develop schizophrenia, but most of the patients who have schizophrenia will not have this deletion. So then there is a sort of a flip-flop, reduces the chances of developing that phenotype, which is a disease. But then there are multiple ways of getting there, because even without this said phenotype, you can still get there. You can still develop the disease. There are other structural variations or CNVs that people have. So how might all of these changes be leading to schizophrenia? So schizophrenia is a defect in processing of information. So for example, the way your brain processes the incoming information to make sense of what is happening around you, that process itself, that perception, is abnormal in case of schizophrenia. So for example, when you're thinking, there may be voices in your head, but then you know that those are voices in your head. For people with schizophrenia, they're unable to make that distinction. To them, that voice in your head is just as real as somebody's difference in processing, is that how the brain makes sense of environment and makes sense of where you are. That perception process is abnormal. So that is the phenotype. Now we have a whole bunch of genetic differences that we see in people who have this problem, this disease. So what is the connection? So one option is that the genetic difference that you see, whether it is a SNP or a structural change, it may be leading to differences in expression of these genes, the expression of the genes where the variation is, or other genes. And then that may then change developmental pathways. So the thing about psychiatric disorders is most of them develop either in teenage or in young adulthood, so teens to your 20s. But then the process that leads to first behavioral change, the phenotype would become evident at that age, but then this has long been believed that the processes, the molecular scale, may be starting at fetal stage. So it could be that differences in gene expression may alter developmental trajectories leading up to young adulthood or teenage, when the symptoms first become evident. So you have the changes against the background, so genetic background, as in the whole genome at large. So it is not just the variation itself, but the background against which it is there that may also have an impact on whether or not that variation ultimately manifests as disease. Adding to the complexity is the fact that everyone who has the diagnosis of schizophrenia, the disease looks differently. So there are variations in how it first manifests, how it progresses. They may have other psychiatric disorders along with schizophrenia, or other diseases, not necessarily related to the brain, but other diseases, for example, cardiac defects, in addition to schizophrenia. Or you may happen that even though this variant that is otherwise linked to schizophrenia, somebody has those, but then doesn't mean that they, this is the thing that but then any of these can intervene, and it may be that even though that variation is there, no disease develops ever. So let's look at how we know that that there is a genetic component to psychiatric disorders. Psychiatric disorders, like I said, they're challenging because there is no overt injury to treat, but then that there is a disease, there is a difference in how those people with the disease are. So how we know how it first started was that psychiatric diseases tend to run in families. Not always the case, but then tend to run in families. So there is people have studied families or twin studies and also adoption studies. So that is one of the first ways that that the genetic component to psychiatric disorders was studied. So here we're looking at bipolar disorder, the genetic risk of that. Bipolar disorder, as the name indicates, there's two extremes of what that disease looks like. You have one end that is mania, where people have excessive energy, they talk fast, they have the think of all you can do with energy. Yeah, you make big plans and all you want to do. So they have excessive energy and to the extent that they don't feel the need to sleep even. So their sleep cycle is disrupted in that stage. And the other end of that is depression. So people with bipolar, in that whichever phase it is, either mania or depression, there are episodes with people differ in terms of how long their episodes are and when and how they manifest. But they will have both of these episodes at some point. So here we have on the x-axis is the relative risk. This dotted line here is chance. So that's a value of 1. So that's any random person off the street. What are the chances that that person will have bipolar disorder? That's it. So that's just random chance. And then any higher number is increased risk. So people who have studied this, so you have, they would obviously, you would take patients with bipolar disorder, and then some of the studies have used controls, which is their match for gender or age, but will not have the disease. So some of the studies have included controls, some have not included controls in their own study, but then they'll use the population, like what are the chances that in this population. So for example, if you're using Indian patients, you'll use the prevalence in Indian population of bipolar disorder as your baseline. And then they have used different patients. The sizes of patients in that group will vary, 200 to 1200. And so you can see all these numbers are higher than 1, which is to say that the disease has a genetic link. When you have people who have somebody who has bipolar disorder, the chances of somebody else in their family also developing bipolar disorder are higher. The chances are higher when it's a first degree relative, which is either parent, child, or siblings. And the risk is lower when it is like a second degree relative or extended family, but then there is a genetic component to it, which is as this figure shows us here. In case of schizophrenia, so this is one, this is another way that we learned that there is a genetic component to this. This is one particular, so like I said earlier, there is more than 100 genes that are linked to schizophrenia. One of them is this gene called DISC1. And this is a fairly large gene. Some of the variants are listed here. These are all within exon 2, as you can see. So there's a whole lot of variation. So when it comes to translating what you know from genetics to a disease, this is part of the challenge, is that you have a lot of genes that are linked to it. Each gene in turn will have a lot of variants linked to it. So going from one variant to say, because this variant is there and that's how this is connected to the disease, that's extremely difficult to prove. But for an example, let's just take one. So this is one change in amino acid sequence here that is linked to this gene, DISC1. So here, this is what's called a pedigree. So my clinical colleagues have to make this frequently when they see patients. So you have, here you're marking which family members have what phenotype, in this case, psychiatric disorder, and which combination of alleles do they have. A line through a symbol means the person is deceased. So here we have, and this arrow indicates, so this would be the first person that the clinician would have seen, which is shown by this arrow and the, and so then once a clinician will interview a subject, then they'll start mapping out, well, how many siblings do you have, or their spouses, and then in this case, children of the subject, and so they would have sequenced the DNA at this site in the DISC1 gene. So this is not sequencing the entire DNA, you're just sequencing a part of it, and to just find out which allele combination that they have. So here we have this person that was seen had depression, but then their spouse has bipolar disorder, it looks like. The clear ones have no psychiatric diagnosis. The orange means generalized anxiety disorder. So you can see that within the family, that people have different psychiatric diagnoses. So there again is your link of psychiatric diseases tend to run in families. One person has one psychiatric disorder, a relative may have the same disease or another, but then you tend to see them together. So in this case, they have tried to map their family pedigree, to saying what diagnosis they have to what genetic or allelic combination they have. So in this case, you see that CT tend to be related more to CT, so nucleotide sequence at this particular site in DISC1 gene tends to be associated more with a psychiatric disorder. But there again, you may not have, it's not always a neat clean of this allelic combination, then therefore this disease. So okay, so those are called linkage diseases. Like you have a genotype that is linked to a psychiatric disease. We have also another way of finding out the genetic contribution, and those are candidate gene studies. So these ones are not, are you able to hear me?
Yes, madam, we can.
Okay, all right. I just want to make sure. The Zoom looked different at my end, just checking. Okay. So the other way are candidate gene studies, which is that you look at individual contribution of a single gene. This is not a very, this has not been very fruitful, I can say, to when it comes to specifically psychiatric disorders. But one of the cases where it has been is OCD. This is one such example. So this here we're looking at this gene is called SAPAP3. And in these mice here, this gene has been knocked out. So what you see here is that when that is the case, these mice seem to show excessive grooming. So to the point that they have like removed, not like pieces of hair are just, their fur is just missing from the body. That's the extent to which they groom themselves. This is part of the symptomatology of a disease called OCD, Obsessive Compulsive Disorder. Here, people people have obsessions, which are negative thoughts or worry, for example, but then to get rid of those negative thoughts, you have these compulsions. So for example, there'd be fear of germs, so they wash their hands a lot. And that, I mean, it is to the extent that then but obviously that behavior doesn't get rid of the thoughts, so it's an endless loop that they get caught in. To the extent that it can be very disruptive. They can wash themselves or clean themselves for hours in a day. So it can be very disruptive for the patients. And that's what you see here. This is over just over a 24-hour period that these mice groom themselves so much they can't seem to stop, to the point of that they cause injury to themselves. So this is one gene. When this gene is not there, you see this behavior. Another element of OCD is anxiety. People can have a lot of anxiety. So these mice also seem to reflect that. You have, so when they're given time to explore, area, the mice with this gene knocked out don't spend so much time that they mice explore a given area, but then these mice show what is the equivalent of anxiety in humans. They don't want to be exploring, they just want to hide in a corner. So so you see both of those, some of the elements of OCD is there in these mice with the gene missing. So that links this gene to this disease, but there again, a lot of information still missing. How do you go from one gene to an extremely complex disease? This table here is from our recent paper, came out a few months ago. So here on the column here, it describes different factors. So like I said, the diagnosis of psychiatric disorders happens by interview. It's a subjective assessment that a clinician does for a patient. So these are the different factors, different boxes that that their symptoms are characterized in. People there can be obsessive about symmetry and arranging. So they can have it. It can be in terms of contamination. So they may wash a lot. They may have mental. The obsessions may be mental. So they have mental compulsions or thoughts that are forbidden, or they can have doubts. They forever doubting that did I close it? Did I lock something? So these are the people that spend a lot of the time checking things, doors, windows, and so on. There is another way is hoarding and collecting. So these are the people who cannot throw away anything because something bad might happen if I throw something. So people with OCD may have predominantly one or a combination. So here what we try to do is we looked at one SNP inside the SAPAP3 gene and try to correlate that the allelic combination at that SNP to see is there a link between the different symptomatology of OCD. And we found that depending people who have the G allele at that SNP, there seems to be a significant relation to contamination and washing. So one specific aspect of the disease that seems to be associated. So here, this analysis, like I said, this is with 200 patients, OCD patients seen at NIMHANS, and it is a statistical analysis. As in statistically, there is a chance that this allele is linked to this manifestation of OCD. These people with this allele tend to their fear is contamination. So their symptoms that they have is that they tend to wash a lot, sometimes for hours. So this is one way that we start with a gene, and now we're trickling down to, well, what are the different variations in that gene, and then how might that help us explain the different symptomatology, the different ways that a disease manifests. Okay, so then how do we, let's now look at the techniques. How do we find out where in the genome the genetic variations are? So one way to do that is what's called a Snip Array. So here what you have, so you start with the DNA from blood sample, for example, and then that piece of DNA is then you sequence a small piece of that. There's different companies. This here are names of companies who make these chips. So you have essentially a chip, a piece that has lots of random pieces of DNA stuck to it. That is these pieces here. And then you sequence, you find out what that sequence is. And then depending on which, when there is a SNP, a sequence change, when you sequence it, it will be the nucleotides that you put on such a SNP are labeled, they're fluorescently marked. So then depending on which kind of fluorescence you see, you can find out which nucleotide is bound there. So this is one way to find out the presence of SNPs in a given sequence of DNA, but this is on a much smaller scale. On a larger, like the entire genome, that is done by what's called next generation sequencing, NGS. So here you start with the entire DNA of a cell. You do what's called a library preparation, which is that you shred this DNA. So that's the equivalent of a single piece of paper, putting it through the shredder. So you have individual pieces that make up that page. So you have these individual pieces. You don't know what their sequence are. So now to find out, you need to have some landmarks. So what you do is you attach, which are these colored boxes here, the green and the purple. These are called adapters. This is small DNA pieces with known sequence. And you attach that to either end of the DNA that you want to sequence. And so during the sequencing itself, you will have, because all of these techniques use the essential principle that DNA binds to complementary sequence. That is, every piece of sequencing, every piece of genetic and genomic sequence uses that principle. So then you have a complementary sequence, a sequence that is complementary to these adapters that you have added. You don't know what's in the middle, but you know either end. So once you start with that, so DNA will bind to this complementary piece and sort of build from there. You use that principle to find out what that intervening sequence is. And once you do that, then the machine that you run this on, the sequencer, the software of that machine will call. So it's called base calling, as in which base is bound, ATGC, which base is bound. Depending on the fluorescent signal it sees. So essentially, this is what is common with Snip Array, that you have all the 4 different bases with different fluorescent signals. So you'll have a different fluorescent signal coming depending on which base is bound. And based on that, then you compare that against the reference sequence, and then you find out whether there are SNPs, indels, or CNVs in the sample that you have. So how does that look for disease? So how do we go from the SNPs, or list a catalog of different variations, to then linking it to design of the study is excruciatingly important. You can't just say, I'll take 5 patients with a disease and 5 will not, and then whatever difference is, that tells me anything. Not really. You need huge sizes, like tens of thousands. That size, I mean, of course, we didn't start there small. At the beginning, they were like a couple hundred and then sort of built from there. But essentially, in whatever size, the study design is that you have, you detect different SNPs, and then you see the distribution in the cases versus the controls, people who do not have that disease. And you look for the distribution. So SNPs are different from disease-causing mutations in that you will always have people who do not have the disease but have the same variant. So in that case, which one do you take seriously and which one do you not? So you do by their distribution. So here we have SNP1 that is there in cases. It's also there in controls and in about the same number. So here we cannot say that the SNP is in any way related to the disease. It's there in both groups. Again, SNP2, here the frequency is different. It is there in more people compared to SNP1. But then when you compare across in cases and controls, the distribution is again the same. So again, not related to disease. But now you look at SNP3. It is also there in both groups, but the frequency is wildly different. It seems to be concentrated in people with disease, not so much in people without. Here you have a positive association. So like I said, you need big, huge numbers of patients, something that a single lab cannot do. So genetic or genomic research rather is done more and more as consortia, which is multiple labs, sometimes across countries that participate in it. This is one such consortium, IC Magic, the Indian Consortium on Mental Illness that our lab is part of. Ours is one of 13 centers across India. And then this is then linked with the international consortium, the Psychiatric Genomics Consortium, the PGC, which is over 800 scientists from over 40 countries, which then all they would have similar protocols to then recruit patients, sequence their DNA, and do the analysis so that you can link samples. So you can do the analysis in bigger numbers and then make sense out of what you see. So this is where we are. We have figured out how we do the sequencing and how we make sense of what that sequencing tells us. So we're at this end where we know something about the genetic architecture. There's a whole different techniques that you use to figure out the functional architecture. So you have a difference in DNA sequence. Does that mean anything in terms of function? So does that change the expression of genes? So for that, RNA sequencing is done. So again, you're looking at across the cell. You're not looking at one or two genes. You're looking at everything at one given time point. What are all the genes that are expressed in any given cell? That's RNA sequencing. You can also variant, like I said at the beginning of the talk, it can be in a coding sequence, in which case you do RNA sequencing, or it can be in a regulatory sequence, which there are different techniques that have to do with chromatin capture. That relies on the principle, like you see here, that when you have a piece of DNA that regulates another piece of DNA, when that regulation needs to take place, there is physical contact of the regulatory element with the gene that it is supposed to be regulating. So at that point, you'll see a physical association. They may be present anywhere in the genome, but then when the gene needs to be expressed, there'll be a physical association between the regulatory element and the promoter of that gene that we're talking about. So that the techniques that help us do that are chromatin capture techniques, which rely on the basic principle that the appearance of the chromatin will be different for genes that are expressed versus ones that are not. So when you have this kind of a thing happening, that chromatin will be open. So you could sequence that bit of DNA and then find out what protein is binding to it, which other piece of DNA is it physically relating to. So these chromatin techniques allow us to capture both. So you can have the promoter, which is where the transcription starts, and the regulator physically associated, and you can sequence the DNA strands that each of them bind to. But of course, beyond that point, you have to go to cellular architecture. What are the other genes that this gene talks to, and which are the cells that express this gene, and at what stage of their development? So in the last few minutes of the talk, we'll go then from variants to genes. So we talked about sequencing. Another such multi-lab consortium called the Psych-ENCODE project. The goal of this project is to fill that gap between, okay, so you have a whole list of variants. How do you go from that to making sense of the biology? So here, they're looking at three genes: bipolar disorder, schizophrenia, autism, and their samples also include healthy controls. So this is where they start. From the previous, you have a list of variations in DNA. So you have a catalog of DNA sequencing variants. How might that be linked to any of these diseases? So one of several ways. This DNA sequence can change which transcripts or how much of a given transcript is present. So for that, you do sequencing of RNA at the cellular level, which is called transcriptome. It may change the level of protein, where you do proteome analysis. You're looking at all proteins present in a cell at any given time. You may do either of those things or both of those things, not necessarily. It may manifest in any of the different combinations. You can have change in transcriptome, but no change in protein. You can have a change in variation that leads to a change in transcriptome, which then leads to a change in protein. Any combination is possible. You can also have where a variant changes the regulation. So one of the epigenetic markers. So this is, for example, like I said, 98% of it is in regulation. And one of the ways that DNA expression or the regulation happens is by methylation of DNA. Some cases, methylation of DNA turns on the expression of a gene. In some cases, it turns it off. So then changing the level of methylation, changing which genes are methylated and at what stage can change the expression of genes, and then change which proteins are expressed. So you can have any combination of these factors happening. And then, so at what extent or to what extent do they play a role in any of these diseases? That's the purpose of this. So they're looking at all of these factors together. So now we have genes or variants to genes. Now, once you know, okay, these are the genes involved, where do you go from there? Do you then look at what are the other genes that this gene is talking to? So genes to networks. So now we're still within a cell at this point, but we're still looking at beyond a single gene. So here we would see, so for example, one is, so you have, so we're here, genetic variation. This can change the transcription, you can change proteins, and this can change the epigenetic marks. So now all of these techniques give us a window into what is happening. When you have this genetic variation, what might be changing? Are we changing the transcriptome, the proteome, or the epigenome?
Dr. Ritika, we are
at
unable
any
to...
given point. Hello? Can you see
me? In between you were lost, so your voice was not audible in between, so I reminded. Yeah, now it's clear.
Now it's okay? All right. Okay.
Yeah,
So
please
start
proceed.
from the beginning.
Yeah.
So once we have a genetic, we're looking at how that genetic variation is, what is it changing downstream? So is it changing the transcription, the protein, or the epigenome? When you do that, when you're looking at any of these levels, we find that the changes that can happen at the level of interaction. A physical interaction of different proteins or different transcripts can change because of a variation, or it could be at the level of correlation. So in that sense, there is not a physical binding, but there is, because you have a change in a transcript on a protein, it can change the functional interactions of what that transcript or protein then in turn binds to. So that is reflected in this figure by either the dark lines, which are the interactions, physical interactions, or the dotted lines, which are functional interactions. Any of those can change. You can also have changes in the development itself. So you have, here we have the different layers of brain tissue. This is right next to ventricle. This is the top surface. And the layers of the genes, the layers of the brain have cells arranged in a particular architecture. So that which cell has to be present in which level, that is indicated by their genes. So their DNA will have instructions on make this cell, and this cell has to be present in this layer of the brain, and then this cell has to make contacts with all of these other cell types, so that's how you go from cellular or changes within cell to how that changes the interaction, changes the interaction of the cell with other cells around it, or change the distribution of how those cells are distributed in the architecture of the brain. And then that can lead to changes in the circuitry that connects different parts of the brain. So starting from here, the radiation, it can affect one or multiple levels. What we get at the clinic is change in behavior, but then we have to backtrack to say, well, what else is changed when you see this behavior? So here we're looking at transcription. So how is transcriptomics, as in looking at transcripts, RNA molecules at the cellular level, how is that done? So I'll just run through here. We have different, so for example, a theoretical example of two genes. You have cells of interest where you see, well, what are the chances that the expression of these genes is linked? Here you have, this is statistical probabilities that the genes are expressed together, or one is affecting the expression of the other. And based on that, your results will look different. So here you see different patterns. a darker color versus a lighter color, indicating different statistical probability of whether one gene is affecting the expression of the other genes. You start with computational predictions. Softwares will tell you that this is more likely to, this RNA will has a site where another protein binds, for example. So if you change the sequence, that might change the binding of the protein. It may then change the fate of that transcript. But then you have to check that whether the prediction that is made by software after sequencing, does that actually hold true? So you have to actually conduct what's called wet lab experiments, where you start with the RNA of the cells in the cells of interest, and then see, do you see binding of RNA binding proteins, or in case of DNA, binding of transcription factors? And then is there change in interactions of different proteins that would have bind to one or more of these sequences? To make sense of the variants, you can see either, there is one or two ways you can go about it. You can start with what's called a seeded approach, as in you already have a list of genes and you want to see how are these genes connected to your whatever disease you're studying. So you start building from each of the individual genes, and you see, well, each of these genes, which other genes are they talking to, and you start to build a network from there. And once your list of genes has grown, because you have binding partners or interacting partners of each of these individual genes, you can then see, well, what are the common themes, what functions perhaps connect these different networks, and then start to see if a theme emerges. The other type of approach you can follow is unbiased. So this is all genome-wide techniques. Why they're valuable is that they are what's called XU, starting with, well, let me see what I see, when the only difference I know is disease or no disease. So in this case, you're looking at the entire transcriptome, and invariably you will see different networks, because different hubs of transcript will be active at any given time. And from there, you start to see, okay, well, what themes emerge? What is being expressed? Is cell adhesion a thing? Is what is happening with actin, microtubule network, for example? What is happening with mitochondrial genes? Those all connect different transcripts. So you see, well, from every expressed, what do I see more of? So that way you label, or what's called annotate, different things emerge, and then you see where your candidate genes are. So you don't start with the candidate gene, they come at end of analysis, and there you see, okay, whatever this annotation is, whatever this theme is, that is where most of the genes that I'm interested in, this is where they are activated. So if there is change in transcription of these, this might be affected, because you have multiple genes in the same network that are affected by the sequence variations. So these are all possibilities that happen once there is change in sequence. So how do we know what's actually happening? So one way we know that is stem cells. Now here, one way to get stem cells is skin biopsy. A hole is punched and you get fibroblasts from the skin and grow out the skin cells. You reprogram those. So you give them transcription factors that turn the clock back on the skin cells, make them stem cells again. But now these stem cells have the genetic material of the person from whom the skin cells were isolated. So you have their stem cells, which you can then convert to brain cells. You cannot know what's going on in their brain, but this is the closest that I get. So in our lab in EBS, instead of skin cells, we use blood cells, but the principle is still the same. The blood cells and then reprogrammed into stem cells and then grow them into different kinds of brain cells. So you can do this in a 2D system, in which case you're growing cells on a flat surface, or you can do 3D, which is essentially a ball of cells. So from your embryology or developmental biology, you'll remember when the cells, after the single cell zygote stage, all these cells do is divide until it's a ball of cells. So essentially what you're doing is growing the stem cells into a ball of cells that will then make the brain. So you have each such individual 3D aggregate, which is called mini brain. So after in an aggregate like this, so here we see this is mini brain or organoid, but here we see what an organoid looks like. And here we see what brain looks like. So there is parallels between the organoid is the closest we've been able to get to a 3D brain so far. So in the organoids, in this particular image, this is cut after 2 weeks of growing it in this form. And what this image here shows that in a control brain, you would have cell division, which is this orange, and the cells, as they divide, they move in an orderly fashion from where they're dividing to where they're supposed to go. But this here is from is an organoid from stem cells from a patient with schizophrenia. So you see there is an obvious difference in how many dividing cells there are to begin with, and their distribution. They're not moving out in this orderly fashion that is required for normal brain development. As I said in the beginning, that people have long suspected that diseases that manifest in teenage or in adulthood have their origins in fetal development, but this is the first time that we're able to visualize that we have these models. That it's indeed that the changes are happening at the fetal stage. I want to show this one brief snippet to close. This is one of our recent disorder, the that Dr. mentioned at the beginning. They're looking at patients with bipolar disorder and trying to. So bipolar disorder, one of the drugs that is commonly used in the treatment is lithium, but then some patients respond to lithium, some don't. So here we wanted to see what is the difference. Is again that also is linked to genetics, and I have to talk that genetic drug response. But this is essentially what we're doing here, is that we have stem cells from patients, bipolar disorder patients. One of them is doing very well on lithium, one of them is not. So these are neural stem cells. This is the cells.
Ma'am, I think the speaker got disconnected.
Audience, kindly stay with us 1 minute. I will get back after talking to the speaker.
Are you able to hear me? Hello?
Now we can hear.
Okay, great.
There was,
so I will start.
There on and off network issue is there from your end.
Sorry, is
okay.
it clear now?
Yeah, now we can see.
Yeah, okay. So I'll just start again, not sure where I lost you guys. So here we're looking at the genetic basis of drug response. That was the goal of this paper here. So we have stem cells from patients who have responded very well to drug treatment with lithium, and one who has not. And here we're looking at differences in proliferating cells. So like I said on this slide here, schizophrenia shows a difference in proliferation. These are the cells that once the brain development begins, there is few sites in the brain where division happens, and then the cells that are created as a result, they have to travel distance to get to their final places where they will then form connections, and which is part of the normal brain development and brain function. So this part is affected in schizophrenia, but we found that it is also affected in bipolar disorder. So here we're looking at dividing cells, and this is person that in stem cells from a patient who has not responded to the treatment, we find that there is actually more. The green dots are the dividing cells, so you see that there is more of these green cells in the cells in the patient as opposed to the control. So the division is more than what it needs to be. So the cell cycle is a function that is commonly affected across disorders. The way it is affected is different, but that this is a function that is affected for sure, that we see clearly here. Then we also looked at mitochondria, which I don't have time to go into right now, but essentially the point is that through the years, about the different ways that diseases manifest. In psychiatry, the diagnosis and progression has so far mostly been confined to the subjective the describing of symptoms by the patient or their family members. But using stem cells, we have a window into what the molecular phenotype looks like. This has, needless to say, important value for us to understand how this disease works and how the drugs that you treat the disease, how they change the behavior of cells. I will just close with that. This is a lab at ADBS. The ADBS includes clinical team at NIMHANS and also researchers at 2 other institutes in Bangalore. So with that, I will close and I'm happy to take your questions.
Thank you so much for thought-provoking talk, Dr. Ritika. I just hand over this session to Professor Shubha Badran to take over question answer session. The session is opened for question and answer. Shubha, please take over.
Yes, ma'am. I'm just checking the chat box.
Okay. The session is opened for question and answer. Kindly post your questions in the chat box, or else directly you can unmute, talk to the scientist. Dear audience. Students, do you have any questions to ask?
There is one question. It says, I want to ask that, as we know that depression is so common between individuals, is there a possibility? I think the question is incomplete. Monideepa, can you complete that question, please?
Monideepa?
Ma'am, we'll unmute her.
Yeah.
Is there any possibility that the people without any SNPs related can also have that disease? Ma'am, I'll repeat the question.
Okay.
We know that depression is so common between individuals. Is there a possibility that the people without any SNPs related can also have that disease?
So without any SNPs is a very difficult proposition to be. I mean, you would have some variations. So everybody, even people within the family, even people who share DNA have differences, and there's polymorphisms are common. So the question is not polymorphism or not. The question is which of those polymorphisms have anything to do with disease. So if the question is, well, of the list of variations that we know so far, if none of them may be there and people may still have depression, yes, absolutely. Because SNPs, just the presence or absence of SNPs does not make the disease. It's also the whole machinery that connects from genetic variation to phenotype. So you may have a SNP but not have the disease. You may have the disease but not have that SNP. Both of those are possible.
Ma'am, another question is, and also can psychiatric disorder be caused without any effect of genetics in it?
Without any genetics, I mean, it will have some effect. So the fact is, like I said, they tend to run in families, but there is always that first person who is diagnosed. So that is not to say that because, so let's say a person A is diagnosed with bipolar disorder. No one in the family has bipolar disorder, which is not to say that there is no genetic component there. This is the person that there is a whole range of from genotype to how a phenotype manifests. There are studies from clinical team from ADBS where they have shown that there is intermediate phenotypes. So person A, let's say, was diagnosed with bipolar disorder, but their relatives will have some of the other aberrations in behavior, not full-blown bipolar disorder, but some aberrations. So then there is a genetic component. It's just not manifesting the exact same way.
Thank you, ma'am. Another question is depression congenital? Congenital disorder.
No, congenital would be something that's manifest at birth. So it's not, most of the psychiatric disorders manifest either in teenage or young adulthood. There can be childhood onset, but then those are, which again points to the genetic basis. And in that case, the what's called as in how many variants there are and how much is each of those then contributing to the disease, that is really high. So the disease manifests much earlier in life, but not at birth. I mean, autism manifests early, like before 5, but then most psychiatric disorders do not.
Participants, any more questions? Looks like there are no more questions, Dr. Ritika.
Okay, well, thank you very much for inviting me. I hope this was helpful.
So now we'll move on to the formal vote of thanks. On behalf of Department of Genetics, Indian Academy Degree College Autonomous, I, Shubha Bhadran, express my immense pleasure to place on record the contributions of all those noble heart who made this webinar possible. Knowledge generated by research is the basis of sustainable development, which requires that knowledge be placed at the service of development, be converted into applications, and be shared to ensure widespread benefits. Today, we witness one such knowledge sharing session. I offer my sincere gratitude to our speaker for the day, Dr. Ritika Sood, Research Coordinator, Accelerator in Brain Disorders Using Stem Cells Lab, National Institute of Mental Health and Neurosciences, Bengaluru, for sharing her expertise with us. Thank you, Dr. Ritika. It was a wonderful session, ma'am.
Thank you. Thank you so much.
I express my profound gratitude to Dr. T. Somshekhar, Chairman, Indian Academy Education Trust, for his constant inspiring mentorship. Thank you, sir. I offer my sincere thanks to Mrs. Bharti, Secretary, Indian Academy Education Trust, for her all-time support. Thank you, ma'am. I offer my humble gratitude to Dr. Srinidhi K. Parthasarathy, Chief Operating Officer, Indian Academy Group of Institutions, for his expert and able guidance. Thank you, sir. I place on record my heartfelt thanks to our beloved principal, Dr. E. Jerome Xavier, for his motivational leadership, guidance, and encouragement in all our efforts. Thank you, sir. I express my sincere gratitude and appreciation to Dr. Savitri Bhatt, Convener, Popular Science Lecture, Indian Women Scientists Association, Bengaluru branch, and BRNS DAE for their contributions and support in organizing today's webinar. I extend my sincere thanks to all the participants. Thank you all for joining us and participating in this webinar. I owe special thanks to Mr. Chandan Rao, Computer and Systems Manager, Indian Academy Group of Institutions, for all the technical support rendered. Thank you all once again and have a good day. Dear participants,